Wow,
over 20 years.
I've seen it 42 from Bon Air, from a mountain. I haven't been there.
Welcome, everyone. It's good to see everybody. Tonight's speaker for the Living World Lecture Series is Professor Gordon Taylor. He earned his undergraduate degree in biology from SUNY Binghamton and a PhD in microbial oceanography from the University of Southern California. As a postdoctoral associate at the University of Hawaii, he was involved in the NSF-funded Vertex, Racer, Adios, and Guanas, I hope I'm pronouncing that correctly, Guanas Basin programs, all involving ocean carbon cycling. His research covers a range of topics focused mostly on marine microbial and molecular ecology, biogeochemistry, carbon cycling, and technology development. He has extensive experience working on anoxia, hypoxia, and eutrophication issues. He was one of the founding PIs of NSF's International Cariaco Ocean Time Series program, studying the impact of climate change on molecular microbial ecology of the permanently anoxic Cariaco Basin in the Caribbean Sea. He has been on the School of Marine and Atmospheric Sciences faculty at Stony Brook University since 1990. Dr. Taylor is currently director of SOMOS's NanoRaman Molecular Imaging Laboratory, where novel spectroscopic and microscopic approaches are being applied to microbiological and environmental questions. Since 2021, he has served as head of SOMOS's Marine Sciences Division. So please join me in welcoming.
Thank you all for coming out.
Yeah.
That's good. Okay. I just finished lecturing for about 80 minutes. I'm already tired of hearing my own voice, and hopefully it'll hold up. Hopefully it'll hold up. So anyway, it's my pleasure tonight to share with you some of the mischief that my group has been up to for about the last 7 years or so. And I want to hopefully stimulate some conversation, and perhaps maybe some folks can see applications that are appropriate to their own research that could be accomplished with collaboration with our group. Okay, so here's my outline. I'm going to be talking a little bit about our motivation after I introduce myself a little bit more, talk about the facility a bit, and then the fundamentals by which these advanced instruments operate. And then I'll talk about some of the applications that we've been developing. Some are, that's right, I can't point at that one. Label-free applications where we don't need any tracers, and those isotopic tracer techniques that we've developed. And then sum up, and maybe there'll be time for a question or two. Okay, so as Maren told you, I'm a microbial oceanographer. My research primarily focuses on marine microbial mediation of the cycling of carbon, oxygen, nitrogen, sulfur. And that's primarily driven by the time that I spent working in the Cariaco Basin, which is off the coast of Venezuela, hence 42 trips over 20 years. And one of the things that essentially constrains advancement in microbial ecology is that most marine microbes defy cultivation. They don't want to live in laboratories. They don't like test tubes. They don't like petri dishes. So we have to rely on cultivation-independent techniques. And I'm going to explore some of those with you tonight. I'll share what we've been up to. So we established NARML about 10 years ago, almost exactly 10 years ago, to develop single-cell techniques that would help us link function with phylogeny, that is, who are the active players in a particular geochemical process, and how active are the individuals. And I'll just make a plug for our facility. We're a campus service center, so we are open to collaborations and users from not only all over this campus, but we have folks come from abroad, from Canada, from other places around the world, and we welcome that. Okay, so microbial food web in plankton is what drives the ocean's biological pump, which sequesters carbon to the deep sea. And the basic food web you see up here, can you see? Yeah, there we go. It's fairly complicated in and of itself. It's basically a food web based on photosynthesis, but there are lots of interactions that we tend to overlook because of the techniques we use. There are many single cell types of symbioses that we know very little about. There is activity around what some authors have called the phytosphere, that is what the environment immediately surrounding phytoplankton cells. We have organisms that can switch back and forth between being photosynthetic and being heterotrophic or being chemoautotrophic. We have saprophytic organisms, types of organisms that Jackie Collier studies, fungal-like organisms that degrade larger organic matter. We have parasitism. We know very little about that. And of course, predation, which is actually what I did my PhD thesis on all those years ago at USC. And we still have a lot to learn about predation. Okay, so most of these interactions are overlooked by the techniques that we use because they tend to be community level, and we're looking at net reactions. Most traditional biogeochemical measurements are looking at net transformations at scales that are really not relevant to microorganisms. And biogeochemistry is really driven at the molecular and individual cell level, and that is dependent on microstructure and cell-to-cell interaction. But most of the techniques we use are blind to these interactions. So it has motivated folks to look at what we call the microspatial seascape ecology. What's happening at scales that are relevant to individual microbial cells. And for microbial ecologists, our holy grail has been to convincingly link the function of an organism with its identity, with its phylogenetic identity, so that we can make better inferences from all the genomic data that is emanating from marine systems now. But many of the current practices are inadequate in capturing intra and interpopulation variability in physiological responses and activity. And there is a lot of cryptic exchange of materials that, again, our techniques are blind to unless we get down to a scale that is relevant to microorganisms. And as a consequence, we are missing a lot, and that affects how well we mechanistically understand ocean processes. So back when I started in oceanography, our perception of what the distribution of microorganisms looked like was something like this, just a bunch of bacteria randomly distributed. And through time, we recognized in the late 90s that at the scale that's relevant to microorganisms, there's a lot of heterogeneity in the distribution of physical and chemical characteristics. So this was a back-of-the-napkin cartoon that Farouk Azam published in 1998. And now we recognize that there is a lot of heterogeneity in the availability of organic matter that supports growth of microorganisms, and maybe a more accurate, better informed picture of what's going on is captured in this illustration by Roman Stalker. That's 1 cubic centimeter, 1 milliliter of water. There's a lot going on in there. There's a lot of heterogeneity, and we're trying to develop techniques that can actually interrogate organisms at that scale. So biogenic debris, so what's that include? Whale poop, zooplankton, carcasses, molting zooplankton as well. That material represents point sources of organic matter, point sources of nutrients, and that contributes to the heterogeneity of plankton. And we've also noticed that suspended particles, organic particles, can have a very different chemical environment. They can be anoxic, they can be acidic, they can have low pHs. And so this contributes to niche diversification on a very, very small spatial scale. Oops. Back. There we go. So the approaches that have been used for modeling, ecological modeling for macroflora and macrofauna have had problems being applied to microbial systems because of our limited abilities to recognize populations. And when I say population, I mean all individuals of a given species within a sample. We have limited understanding of their function and their physiological constraints, and we have almost no good information about variability between populations and within populations out in nature. Okay, so I like this illustration. It shows what a bulk analysis type of approach gives us. It gives us an average. So a single bar, if we do a bulk analysis. But if, do I have a pointer or not? Anyway, okay, so if we do bulk analysis, we get a mean mean value for some phenotypic trait. But if we start individually interrogating cells, we get a distribution that looks more like this, where we have different responses. And in this figure here, is the mean meaningful in 2 out of 3 of these? The mean value for some sort of phenotypic trait has, in the upper panel, no occupants whatsoever. It's only if there's a Gaussian distribution that does the mean mean anything, right? So without drilling down and looking at individual cells and behavior within populations, there's information that's lost. Okay, so to address some of this, we in 2014, we established this facility in Selmas with a NSF grant, a major research instrumentation grant. And in it, we have a Raman microspectrophotometer and an atomic force microscope. And as I mentioned, we have all kinds of clients that come in and do really interesting things. Raman can be used for material science, for biology, for chemistry, and biomedicine as well. The instrument looks like this. It sits on an optical bench, and parked right next to it is the working head of an atomic force microscope. We actually have accessories that allow us to optically couple the Raman microscope to the atomic force microscope so that we can actually direct laser light down to the cantilever tip of an atomic force microscope and get chemical information as well as topographic information from the atomic force microscope. It's a rather unique capability. So what is Raman scattering, you may ask? Well, it's not this. It is something a little bit different, and it's spelled differently as well. Raman is R-A-M-A-N, at least Raman I'm talking about. So basically, it's a technology that was first predicted in the 1920s by a German chemist, but it was proven by C.V. Raman in India, in Calcutta in 1928. And essentially, what Raman scattering is you irradiate your sample, and it's usually non-destructive, by the way, so we can look at things without destroying them. You radiate it with laser light. It's monochromatic. It's a single wavelength, single energy. Most of the light is Rayleigh scattered at the very same wavelength, same color. But 1 in a million, 1 in 10 million photons collides with a molecular bond, gives up some of its energy, causes the bond to stretch and vibrate at a characteristic frequency, and those are the Stokes-shifted, keep forgetting, the Stokes-shifted photons, which is what we measure. This is what Raman measures. So it's basically the shift from the laser line, the Rayleigh line here, to whatever energy level that the chemical bond absorbed. Sorry. So this is an illustration of one type of vibration that's set up by laser light, and that would give off a characteristic peak that we use. The simplest example of a Raman spectrum is from the diamond. In fact, Raman scattering is used to grade diamonds by diamond merchants. And basically, it's a carbon crystal lattice. It's all the same atoms with the same bond lengths, with the same energy. You get a single peak because there's a single energy. But if you look at more amorphous, complex material like a bacterium, you get a much busier spectrum. But from that spectrum, we can extract information. We can extract information about presence of nucleic acids, certain amino acids, carbohydrates, lipids, etc. And this is the phenomenon that we take advantage of. And as I said, it's a non-destructive technique. So in theory, we could look at living organisms with this. Typically don't, but we could. What's atomic force microscopy? Well, it produces images that are not unlike scanning electron microscopy. So this is an individual E. coli cell that was produced in an atomic force microscope. And basically, it produces this image using totally different physics than any other type of microscopy. It's a scanning type probe microscopy. It has, in the horizontal, a 0.4 nanometer resolution. In the vertical, it has a 0.01 nanometer, that is a 0.1 angstrom resolution. And for reference, a human hair is 1 million angstroms on average. And so we're detecting differences of about 0.1 angstrom. So this allows us to get images of many things, but we're focusing on cells. And that image was taken under conditions we're sitting in right now. Not high vacuum, not dehydrated, not only preserved. The only requisite is that the cell can't be moving around. It's got to be stationary, but it can be totally hydrated and preserved. So we can get sort of undistorted images of organisms that, if we used electron microscopy, may look a little bit different. This is a comparison of an SEM image of a diatom frustule. That's a silicon dioxide shell. Diatoms live in glass houses. Here's a living one right here. So SEM, atomic force microscopy. I would argue that you get slightly more information out of the AFM image than you do from the SEM image. And this is an image that Tanya, our technician, produced with a colleague from Mississippi, and that's a close-up of a diatom frustule produced in our lab. Okay, just to show the power of AFM, that is a double-stranded DNA molecule. There's 2 of them, actually. And so we can image DNA under ambient conditions. These larger images are actually artificial plasmids that were produced by a colleague. And so what we're looking at are tiny bits of DNA imaged under environmental conditions. Okay. Advanced. All right, so let me go to some of our label-free applications and to power through those. So there is a lot of concern that when you do an incubation and you add a tracer and you alter conditions, that maybe you're creating artifacts. So there's a big push for techniques that are label-free, where you haven't done an incubation. And Raman is one of those approaches that we can use. There are, this is the Texas brown tide. This is the vegetative cell that actively reproduces and makes these harmful algal blooms off the coast of Texas. And it turns out that these organisms form resting spores, which will basically sit in the sediments and wait for conditions to get better and then germinate. Previous publications had identified what they called a red body. They had no idea what that red body was. We did a little Raman spectroscopy on it and determined that the red body is actually sterols. And so now they're called sterol droplets in the literature. This is a paper we had with Chris Goldler and a former student not too long ago. Oops. Okay, so I'm putting this slide up here to illustrate how we get spatial maps of chemical characteristics of cells. So basically, on the computer monitor, we set up a grid, and we decide, okay, we're going to take a spectrum every 1 micron, and we're going to raster back and forth, back and forth, back and forth, and produce a 2-dimensional image. But we can take it a step further, and we can do multiple focal planes and get 3-dimensional images. So this is basically we're taking a spectrum at each node, and our resolution is basically how fine a resolution do we need to see the features that we're looking at within a cell, how much time do we have, how much can we afford. So it's trade-offs between all those considerations. And we use 2-dimensional mapping to look at intracellular storage products. And in this case, I'm showing an example of Tetraselmis striata, which is a green alga, and it is being heavily researched for biofuel production. And what we were able to do is map out the distribution of starch. And the upper panel, this green sector, is the starch deposit that the cell has created. DNA is orange, and the blue around is protein. So you can do these 2-dimensional maps on cells. Well, there's your scale guards, 3 or 4 microns in diameter. Okay, basically that's the same. We've done time courses. This starch probably is a reaction to nutrient limitation. When cells run low on nitrogen or phosphorus or iron or some other limiting nutrient, if they have sunlight and carbon dioxide, they'll store starch as energy reserve for better times. And this is basically a time course incubation, and the magenta is the starch deposit. We start out with no starch when they have all the nutrients they want, and then 3, 7, 14 days are almost all starch because they run out of probably phosphorus. So this is one thing we can learn. We haven't added any tracers, we've just grown the cells in culture, but we can take samples from nature and make the same kinds of observations. Okay, there we go. Bacteria, this is a very, very much an enlarged bacterium, have the equivalent of fat storage. It's polyhydroxybutyrate, PHB, and that's shown here. Basically, the intensity is proportional to the height of that peak right there, and the blue is the intensity of a peak for phenylalanine. So we can unambiguously see the amino acid phenylalanine, and we see that there's substantial storage of this PHP. which is equivalent to a lipid storage product. They're getting fat. That's what's happening. Okay. I think this is the last illustration of a two-dimensional label-free application. This is the Coccolithophora emiliani huxleyi, cosmopolitan. We find it from the equator to the poles. It is a big player in marine carbon cycling because of its calcium carbonate plates, those coccoliths. And in this case, Tanya has mapped the distribution of protein in blue, the distribution of fully saturated fatty acids in purple, and the distribution of cholesterol in red. And we can learn something about the physiological state by looking at the relative proportions of those. Okay, so from the label-free segment of my talk, what the takeaways are that basically we can get physiological information without stains, without tracers. We can do this again in natural samples that we just pluck from the ocean and preserve, or in lab cultures. But this is an example of culture-independent techniques that we can employ. And basically it allows us to look at intracellular storage products and reveal whether there's nutrient stress or not. Okay, now in techniques that we've developed, we use stable isotopic probing, where we do introduce a stable isotope. And I'll give you two examples. One is carbon-13 and the other is deuterium. But first, a little primer on how this works. We have what I refer to as the neutron factor. If you have two carbon atoms with a single bond and they're both C12, their vibrational frequency is shown up the top, 1157 wave numbers. Whoops, I was pushing the wrong button. No wonder. Okay, so reciprocal centimeters are wave numbers. They equate to frequency of the photons. So if they're both C12, they vibrate at that frequency. If one is C13 and one is C12, they vibrate a little bit slower, but that one extra neutron slows down the movement of the two atoms. And if they're both C13, the frequency is 11, 12 wave numbers, and the vibration is even slower. So we can detect this on single cells, and we're seeing the effect of a single neutron. This is phenylalanine, which is one of our diagnostic peaks that we see unambiguously in all our biological Raman spectra. And if there are no substitutions, we have a frequency from phenylalanine at 1002 wave numbers. If there are two C13 substitutions in the phenyl ring, we now have a vibrational mode at 987. If we have four carbon-13 substitutions in that phenyl ring, we have a slower one, I forget what the number is. And at 968, if we have all carbons substituted with C13. So with this neutron effect, we can actually use, as I jokingly say, as a single cell mass spectrometer. Okay. So our first demonstration of this was to take Synechococcus, which is a cyanobacterium, a second most abundant photosynthetic organism on the planet, throughout the world's ocean and in fresh waters as well. And we tried a series of enrichments of isotopes. So the serum bottles are just showing you how much C13 enrichment there are. From natural abundance, 1% C13, we enriched up to 54%. And just to convince ourselves that adding this amount of isotope didn't lead to an artifact, we measured the population growth in all those treatments, and they're indistinguishable. There is no difference. Their growth is not affected by additions up to 54%. If we went above that, we might see something different. But up to 54%, we're good. And in theory, if if we had the medium was 100% C13, at the beginning of an incubation, we'd have a cell that was 1% C13. After one generation, n equals 1, they'd be almost 50% C13. After 2 generations, 74. After 3 generations, 3. So we typically, we don't enrich 100%. We enrich up to 40% or something like that. But this basically shows the kinetics of how rapidly the cells would become heavy. So what I'm plotting here is the number of generations against the fractional abundance of C13 in the cell. And this is all predictable thanks to Marin's husband, Steve, who does all our math for us. Basically, we labeled cells we did time courses, and we labeled at many different levels. And what I'm plotting here on the right is the frequency shift, what we call the red shift, lower frequency, red, warmer color, lower energy, at the 3 diagnostic peaks for carotenoids, and they all show a very tight response, tight linear response, and each one of those circles is the mean of 25 randomly selected cells from the culture. We think that our detection limit for this assay is somewhere on the order of about a 3% change in its carbon isotope is detectable unambiguously. Okay, so I'm not going to go through the equation. Basically, I'm only putting this up to let you know that the end product that we were after was the specific growth rate of single cells. So that's mu sub SC, single cell. And from knowing how isotopically heavy the cells are, how much time has transpired, we can in fact calculate the growth rate of each cell in our sample that we interrogate. And to convince ourselves that it wasn't biased by how much C13 we added, we took subsamples from all these enrichments from natural to 47%. We we basically took aliquots from each of those, mixed them together randomly, put them on a slide, and this is the autofluorescence images of each of those cells. And the value on the left is the single cell growth rate. The value in parentheses is the population growth rate, determined by fluorometry of the entire population. So we're seeing complete agreement between sort of a standard bulk measurement for population growth and looking at what the individual cells are doing. Oops. Okay. So what surprised us in this study was that we were seeing about a coefficient of variation in the population of about 27% from cell to cell to cell. And we worried, is something wrong with our assay? Is this an artifact, or is it real? And we did a similar experiment with the diatom, and we found significant variability. And I started looking at the literature where people were doing single cell studies using totally different orthogonal techniques than what we were using, totally different. And they are reporting for bacteria and diatoms coefficients of variation of 19 to 51%. In clonal cultures, came from one parent living in a flask. What could be more uniform than a flask of seawater media and genetic stock that came from a single parent? Still seeing in these isogenic populations 20, 30% variability from cell to cell. Not just our technique, but other techniques as well. So that's kind of interesting that there's a range of growth phenotypes in these isogenic cultures. And so I have to ask, if we're seeing this in a uniform environment, in a flask that's being shaken and illuminated perfectly, everything just right, what's really going on in nature? How variable is cell to cell and intrapopulation physiology? I suspect it's really high, but we have very few ways of appraising that idea. Again, back to our little friend, bulk versus single cell. Okay. So that was for photoautotrophs, photosynthetic organisms, phytoplankton. We asked whether we could do something similar with bacteria, using a different diagnostic peak than we used for phytoplankton. So we used everybody's friend, E. coli. It was convenient. And we grew it in a C13 enriched organic media over 160 minutes. And as I mentioned before, our diagnostic peak is phenylalanine. So at the beginning of the experiment, we had a very strong peak at 1002. That's isotopically light, phenylalanine. And then through time, we saw that peak practically disappear, and the one at 967 grew as phenylalanine got heavier and heavier. So we went through the same math, the same exercise to get growth rates, and the shaded bars are growth rates determined for the entire population just using optical density, spectrophotometry, the whole community, and the lighter bars are mean single-celled measurements based on this phenylalanine shift. So they're statistically indistinguishable. Our single-cell rates have bigger error bars, but those aren't really errors. That's natural variability. It's not an analytical uncertainty. It's that there's a lot of variability in the members of that community, that population, rather. Okay. All right. So I mentioned the one other tracer that we've been using is deuterium. And basically, a few people have, I'm not first to think of it, a few of my colleagues have been tossing it around as well. And we see it as an agnostic assay that we can use to detect biosynthetically active organisms. And it doesn't matter how they earn a living. It doesn't matter whether they're a photosynthetic organism. It doesn't matter whether they're chemoautotrophic or chemoorganotrophic. If they're producing macromolecules, they're consuming a little bit of water. And if you put deuterated water in there, they're going to get isotopically heavy. And the signal is strong. So this is a control. This vibrational peak here is the stretching mode of hydrogen and carbon. And if we incubate cells with C13, deuterium, deuterated water, a peak appears where there was no peak. And so this is another unambiguous indicator of isotopic enrichment as cells are biosynthesizing. So we can come up with an assimilation rate by just looking at the ratio of the area under that peak compared to the area under the sum of both of those peaks. We get an assimilation rate. And from this, and this is basically, again, from single cells. And also, it doesn't matter whether the organisms are aerobes, anaerobes, fermentative. This technique works for all of them, or at least it should. Okay, so this is a little bit of work that my student Natasha has just produced here in the audience. And this is employing the deuterated water technique to the field. And basically, this is in the Eastern Tropical North Pacific in an oxygen minimum zone. And we've sampled in oxic waters and anoxic waters. And it's just looking at the distribution of isotopic enrichment. So the vertical line is the threshold. To the right, we think there's biosynthetic activity. To the left, we're not so sure, because the kill control basically falls. Most of the points fall there. So each one of those points is a single cell. And we see that the average level is different from depth to depth, and the distribution of activities changes. So we are, on our to-do list is to convert these fractional abundances into growth rates, and also to employ fish no fins, no scales. It's fluorescent in situ hybridization. So we can use phylogenetic probes to determine who are the active guys and who are the lazy ones that are not doing anything. So we can use genetic probes to link function with phylogeny. All right, moving on real fast. Looking at mortality in bacteria. I had a postdoc, Felix Weber, who did a series of experiments where we had control that was in C12 media and enriched at C13 media, and basically grew them up so they got isotopically very heavy. This particular strain of E. coli is UV sensitive, so we can stop it from growing and then watch it disappear when it gets eaten by a predator. Our predator in this case is freshwater ciliate, Tetrahymena piriformis. We start with an azenate culture that's grown on dissolved material entirely, and then we introduce Tetrahymena to isotopically heavy prey and isotopically light prey. And we fix samples, we count cells, both types of cells, and we do Raman. Same kind of analysis with the Raman. We look at the shift of the phenylalanine peak from the beginning to 2.5 days out, almost 3 days out. And this is the entire spectrum. We're just looking at where phenylalanine falls. This is a DIC image of one of those Tetrahymena, and you can see the food vacuoles very nicely. Those are individual prey items. Those are all individual bacteria within a food vacuole. These are fresh food vacuoles. They just endocytosed the bacteria, and as the food vacuole matures, the food gets digested and starts to empty out. Okay, so we did this growth experiment and took advantage of our ability to do two-dimensional mapping. And this is taken at 6 hours, and the warm colors show where there's isotopic enrichment. The cool colors show where there's yet to be any isotopic enrichment. That's at 6 hours, and those are probably the food vacuoles, chock full of E. coli that's heavy. After a little more than a day, the predator is completely labeled. It's basically in equilibrium. It's equilibrated with its prey, 85% enriched. So then we ask the question, can we do this in 3 dimensions? Can we look at these delicate cells that have no cell wall, rather than dry them and flatten them on a slide? Can we look at them fully hydrated, just preserved, never been squashed, never been shrunk? And so we do have a water immersion lens at 63x magnification. And Felix got real lucky. He deposited his fat Tetrahymena onto a slide, and they stuck and stayed in the same place for 13 hours, totally by accident. We didn't do anything intentionally. And then we basically did 3-dimensional mapping. And in this case, we're just looking at the distribution of macromolecules. And we have 7 focal planes separated by 4 micrometers each. And it's not a trivial task. It took 13 hours, but you set it up before you go home for dinner, and you let it run overnight. It's all automated, so you don't walk away from it. Okay, and our software is pretty cool. Capture the image, and in this case, we're looking at the distribution of DNA in red, protein in blue. Now, if you're really paying attention, you will see that there are more than a single red spot for the nucleus. And that's because ciliates have micronuclei. They can have as many as 20 micronuclei. You know? They can have a lot of micronuclei. So there's DNA that are in these little satellite micronuclei, as well as the macronucleus. And so we're able to image all of those. At first I was thinking, oh, maybe that's the bacteria in the food vacuoles, but this particular individual never saw bacteria. It was grown in dissolved media that had no bacteria in it. Okay. So we can take that same approach and do a stable isotopic probing experiment and show this was 6 hours, I believe, where most of the isotopic enrichment is. So this is another one of those tetrahymena cells, and we're looking at it in 3 dimensions. The warm colors are where it's isotopically enriched, cool colors, no enrichment. And the other interesting observation that Felix and Tanya had is there was a void here where the only thing that showed up was a spectrum for water. It's internal to the cell. I had to think about that for 2 seconds. Freshwater protists have contractile vacuoles for osmoregulation. So there are vacuoles that fill with water and then they pump it out because they're hyperosmotic with their environment. So the water goes in, they make these contractile vacuoles, capture excess water, and then squirt it out. So we think that's what we captured there. Okay, last part of our story is looking at another avenue of mortality, and that's viral infection. And we continue to work with E. huxleyi. As I mentioned, it's really an important component of phytoplankton, especially for carbon export because of those coccoliths. Satellite photograph of a spring bloom off of Great Britain to the east. And we have in culture one of the viruses that attacks E. hux. And so Elena, who's here in the audience, and Tanya have done a bunch of experiments with these guys. First thing I wanted to show you was AFM image of purified EHV 163. So it looks like popcorn, but they're actually 160 nanometer diameter enveloped viruses. Here's a single one, and again, an average diameter is about 160 nanometers. That's the AFM image. This is a transmission electron micrograph of a different individual, but the same virus. And so it's an unusual virus that attacks phytoplankton because it has a lipid envelope. Most microalgal viruses are naked, they're just protein shells. So this is kind of an unusual guy. We asked, can we actually get Raman information from viruses? The answer is yes. So that little spot there, maybe it was a single virion, maybe not. But these are spectra of 20 aggregates. The black line is the average for each pixel. The shaded area is the standard deviation around repetitive measurements from different individuals. So we had aggregates, we had the average of 5 individual virions, and here is spectrum from a single virion. It's 160 nanometers in diameter. We have signatures for DNA, we have signature for phenylalanine right here, 1002, and we have signatures for lipids for their envelope. Okay. We observed something that was kind of curious. A number of the particles that we interrogated didn't have the typical viral spectrum, the complete spectrum that you see in the middle here, complete virions. Absent from those spectra were the modes for DNA, the protein mode, the phenylalanine, but there was plenty of signature for the lipids. So the lipids are over here, and when Elena did a number of surveys by electron microscopy, she found a lot of ghosts. This is either an empty envelope or a cell vesicle. Do we know yet? We're in the process of studying cell vesicles. So the individual particle, again, was missing DNA, was missing protein, and so we were able to resolve that from the spectra. one of the things that really surprised us is when we did two-dimensional mapping of EHUCs. Again, these are like 4 or 5 microns in diameter. Healthy EHUCs had DNA in red, protein in blue, and if we stripped out just the protein signature or just the nucleic acid signature, we had a fairly condensed nucleus contributing to the DNA. But post-infection, just 4 days after infection, we saw that the DNA signature was more dispersed. And these electron micrographs show a healthy EHUC cell, and this one shows all those little black dots are individual viruses. The nuclear membrane is gone. And so all these little packets of DNA are dispersed throughout the cell. So we have TEM confirmation of what we're seeing with these DNA portraits of Raman. So we took that a step further and we looked at many cells before infection, day 2, 3 days after infection, and each column, the blue column is protein portrait, the red is DNA, and the numbers are essentially the integrated pixels that are occupied by a signal from either DNA or protein. And we compared pixel intensity over the course of the incubation, and protein, not a significant trend, but the DNA, we can see, significantly decreased. It became more variable as the infection progressed. Okay, I think we're in the home stretch. So we took EHUCs, we grew it in regular media, we grew it in media that was enriched with carbon-15. So we now have a host that's isotopically heavy, and then we infect it with the virus and ask the question, does it inherit the isotopic signature of its host? Well, obviously it should, but can we demonstrate it? So basically, this is the full spectrum of the host, full spectrum, full spectrum of the virus, and then this is just a blow up of the region where the phenylalanine is, and from the relative heights of those peaks, we can calculate what the fractional abundance of C13 is in the host, 0.65, so 65% enriched. We look at the virus from that host, 0.63 or 63% enriched in C13. We can actually detect isotopic enrichment in the viruses. Maybe this will be useful someday in experiments where we want to know where that virus come from. Did it come from an isotopically heavy host? Okay, wow. So what I want to message I want to leave you with is that these approaches have fairly relaxed sample preparation requirements. We do AFM under atmospheric pressure. We don't have to desalt. We don't have to sputter coat. We don't have to analyze under high vacuum. We can do it on the desktop right here tomorrow. With Raman, we can look at live cells. We can look at frozen cells. We can look at formalin preserved, which is what we typically do. So our sample requirements are very, very lax. I mean, we can do isotopic analysis, essentially use the Raman as a single cell spectrometer. This is an approach, the Raman is an approach where we can look at phenotypic variations within populations and phenotypic variations among populations in culture and out in nature. And when we couple Raman with stable isotopic probing and we add fish to the mix, we can then directly link function with phylogeny and get activity measurements along the way. So Basically, these tools are giving us a window into the microcosmos, allowing us to look at some of this microspatial heterogeneity that eludes most other techniques. And that should permit us to look at pathogen-host interactions, symbiotic and parasitic interactions, and we can do it in 2 dimensions and 3 dimensions, and then if you do a time course, there's your 4th dimension. And last point, only limitation, imagination, time, and money. Okay, so I want to acknowledge Team Narmal, Tanya and Elena, who are here. Liz Suter, who was one of the first students to start using Raman in my lab. She's married to Luis, who does microplastics using this instrumentation. Former postdoc Felix Weber, co-author and friend Steve Beaupre, who does all the math. Another former student, Ashley, come on. You know her?
I did.
Okay, yeah, Ashley. And Natasha Lukovic, who's here. Anyway, thank you for your attention. And if you're not totally worn out, you can ask me questions. I'll be happy to answer. Maren.
So you mentioned one of the limitations is money. How does the cost of this tool compare with other imaging techniques?
It's cheaper than most. So our competing or complementary technology is nanosims. So that's nanosecondary ion mass spectrometry. The service contract, the mandatory service contract for that is $100,000 a year. My service contract is 0. Purchase price was probably a 10th of what nanosims is. Yeah. So cheap. And AFM, our only expense are the tips. Now they're quite delicate and they break pretty easily, but we don't have a maintenance contract on that either. So it's basically our primary cost is labor. Gotta pay time. Jeffrey.
Questions I should have asked you 5 years ago.
You mean before you retired?
No, before when I was trying to get Robin to work for me, but it didn't. You have these spectra you show, and then you have a spatial map. Here's DNA, here's RNA. So I know from that you can tell DNA from proteins and other things.
Yeah.
Can you do anything within those spectra to know something about the composition of the DNA? So it's just DNA. So you can't sequence it, you can't look at
There are
different types
people
of.
looking at that, and the way they're doing it is with a similar system to what we have, coupling AFM with Raman. And essentially they're putting a strand of DNA on a microsurface on the AFM stage. They have to have the alignment just right so that they can move the cantilever tip along the axis of the DNA. You get a spectrum every, I don't know how many nanometers, but you will get a spectrum of the individual base pairs.
I actually had a friend who's now dead who used AFM and constructed sequences in there, Ned Seaman. And he made walking DNA and all sorts of other things. He made cubes out of DNA. He's a very famous guy. He made structures.
Who is that?
Ned Seaman. He made structures of the cubes and so on, tetrahedrons and so on. And they even tried to make a computer out of DNA with that technique, with AFM. But I don't know, but they actually constructed sequences they put in and analyzed it. But I don't think they could sequence anything complicated. So that can't be done as of yet.
So I was early on, early on, I was actually a shill for one of the companies at Harvard. They were doing a demo at Harvard, and they were selling basically the coupled instrument that I have. And there was a group there that were talking about using a coupled instrument to do long reads, very long reads, because you don't need to chop it up like you do for sequence.
Because that would certainly be a holy grail. If you could do that, you then could really do very interesting experiments.
But now the nanopores out there now, and what's the read length on those these days? A few thousand, right?
It's tens of thousands.
Tens of
if
thousands.
I mean, the limitation is the extraction, not the sequencer at this point, in getting long reads.
What do you mean by extraction?
Is that you often fragment your DNA if you're not
Yeah.
gentle with it, and so that the sequencer can sequence a very long fragment, but if the DNA itself is fragmented, you can no longer.
Right.
And so you have to use some gentler methods. You can't be
In the back.
Piggybacking on that question, well, maybe not for the sequences, but can you use this technique to like infer in terms of nucleosomes in eukaryotic cells? Like basically, can you identify like compact regions of histones or something like that?
I think so. I don't know why one couldn't.
Well, that's pretty cool. But I just wanted to know that part. The other one is like when you mentioned the diatoms, right? Like there's a lot of variation.
Oh, yeah.
You mentioned there was a lot of variation. Do you feel like is it possible to, I mean, I am assuming that there is a non-stochastic explanation to that at some point. And I'm thinking, well, maybe it's like very local, variation in resource availability around the cell or something like it.
Right.
My question is, you mentioned that it could be possible to put time resolution there, but also can you use any of these combination of methods to get information about the immediate chemical surrounding, like chemical environment around the cell? Is it possible, or it has to be like processed by itself?
Yeah, Raman is not super sensitive. So if you're talking about monomers and small peptides and things like that, if they're not in high concentrations. So it's outstanding for macromolecules, condensed media, and so on. But there are tricks you can play with Raman. There's surface enhanced Raman, there's tip enhanced Raman, there's resonance Raman that amplify the signal by orders of magnitude, but also potentially introduce artifacts as well. Chuck?
Tell me about the spectroscopy. Do you have to average some number of spectra to produce the spectra that you're showing us? Is that the reason that the bacteria need to be not moving? Is it, now I'm going to ask another question, is it possible to develop software that would track them so that you could obtain spectra or images from active?
I think that's
bacteria?
possible. The stage is automated and it has 100 nanometer step size in 3 dimensions. So it's all in the computer's control, and it might be possible. But many of the spectra I showed you were single acquisitions that took seconds or fractions of seconds.
You can't see a sequence. I'm just thinking, listen, could you observe fertilization in real time with this technique? Simple question.
Yeah.
Because there's a point at which a sperm head is immobilized. As one sperm, the whole surface is immobilized and changes its character. Could you see that sequence of changes in time, which would be a matter of a tenth of a second?
Well, we were just looking at a paper, an AFM study that showed a healthy E. coli, and then an E. coli after viral infection showed AFM and it showed viruses outside of E. coli, and then sort of the decimation of cytoplasm as the viruses were reproducing.
I mean, you could image cell division.
I think there's better microscopic
Do you mean in provinces or like on a different cells or same cell divided right
It's
now?
the same cell, I think like with fertilization, there's a sperm entering and immobilizing that membrane, which is very famous, but an inferred reaction. I don't believe anybody has any direct observations of that.
I don't think, because if you have a cell, you have to map it. And mapping for the cell like a HAX or tetracellular is about 2 hours.
Yeah, so we're not taking
To do the map,
Yeah.
to do the case.
Oh, so
To
what
do
you want?
all the spectrum, which, like, each point, like 1 micron by 1
Oh,
micron.
yeah, I'm talking about something that happens in a tenth of a second.
No, for some materials, yes, it can be done, but here we have very little of material, ramen is not very sensitive, so you have to irradiate it for a rather long time
I see.
to get the answer, so,
Oh, I see.
to accumulate your.
I see, to get the analysis,
Yeah.
you need those radiations.
Yeah, it's not a snapshot, you're getting a single point.
You're
every few
rastering
seconds, and you're
the
rastering,
cell,
yeah. The stage is rastering under the objective lens.
and that takes 2 hours or so.
Okay. I
Yeah,
see.
depends on the size and the resolution. Right.
But for example, if you want to see something something goes on the same point, so then you can focus at this point and take spectrum like every 10 seconds, for example. If the something is quite good in ramen, and then you will see the changes.
So single point beam time series, images,
What's the dimension of the laser?
About half a micron, like a little bit, depends on the laser, so the shorter
Yeah,
or
it's
longer,
wavelength dependent. So if you were to use blue,
it will be very small for
it'd
it.
be quite small, and the focal depth would be about 300 nanometers. So the actual sample volume would be 300 nanometers deep, and maybe a half micron across. Yes?
How about the spectra being identified or not with specific molecules? Like, is there every molecule has its own spectra is known, or is something that's expected? That
Many are known, but the problem is if they're, the spectrum that you get in cells in particular is, what you're seeing is a contribution of multiple compounds. And there are very few that have just one signature, like phenylalanine. And the deuterium carbon stretch are unambiguous, but there's lots of overlap. You have very broad peaks, and underlying those broad peak, a broad peak may be multiple peaks at different frequencies.
in this 3D mapping, like how is it that you can identify one level? Let's think distinctly from levels that are stacked.
Well, practically the way we do it is we determine how thick the cell is, and then decide how many focal planes we want to resolve the features of interest. And we get the thickness just by focusing from top down to the substrate and looking at ramen intensities, right? So we have top and bottom, and we subdivide it.
These cells which we did, they're quite transparent.
Yeah, if you're opaque, forget three-dimensional mapping. But most cells are amenable. And then the other limitation that we struggled with that I didn't bring up is if you have pigmented specimens that are autofluoresce, you hit it with a laser, and all you get is fluorescent, broad fluorescence with no prominent information underneath. So Elaine and Tanya come up with a method where we can suppress autofluorescence from chlorophyll and carotenoids and so on. And basically repress fluorescence irreversibly. So once we treat the cells, we can analyze all of them in one fell swoop, one fell swoop. And that was a big step forward because folks in the past would just photo bleach every target. Now it takes minutes and minutes, and then you want to do a map plot, it really adds up. But pre-treated samples where all the cells on a slide have been, their fluorescence has been quenched with peroxide and high intensity white light, they don't recover the fluorescence. And we get the same signatures. We've done controls with E. coli, we treat it the same way as we treat it for phytoplankton cell. And we get the exact same stuff after that treatment. That was a big hurdle. Fluorescence. Yes.
A question that is perhaps part of the imagination for him, perhaps not a poignant question, but has anybody captured neurons at this level?
Again, I think you've got an issue of it's a rastering type technology to do too much. I think super resolution microscopy and confocal fluorescence might be a better tool. I don't know.